Hello there. In this set of slides, we're going to see animal mitosis as it appears in whitefish embryo, and we're going to take the phases in order, beginning with interphase, then prophase, metaphase, anaphase, telophase. All right, let's begin with interphase. You say, what am I looking at here? Well, here's the nucleus. You can't see any chromosomes. Here's the outside of the cell, like so. This cell is in interphase. So is this one. There's a nucleus right here. Here's the outside of the cell. You can't see any chromosomes in the nucleus because they're stretched out. They're uncoiled. You can't see them. So those two are interphase. Now we have a prophase. And so the chromosomes are still kind of toward the center of the cell, but now you can actually see them because they've coiled up. They look like little whiskers, perhaps, or little worms or something. And so this cell will be in what phase? Prophase, the beginning of actual cell or division of the nucleus, beginning of mitosis. So here's the outline of the cell out the outside of the cell. Prophase. And then another prophase. Prophase, again, here's the outside of the cell out here. Here are the chromosomes. We can see them now. They look like little whiskers or little bitty worms or something. And they are coiled up to where we can see them with this type of microscope that I use to take these pictures. What do we have now? Ooh, we've moved on to metaphase. Those coiled up chromosomes that we can now see have been grabbed by these spindle fibers here, spindle fibers here, spindle fibers here, and taken to the equator of the cell, the equatorial plane of the cell. What phase is this again? Metaphase. Here's another metaphase. Chromosomes all at the equator of the cell. Here's the spindle fibers. Metaphase. Here's another metaphase. Chromosomes at the equator of the cell. Here's the spindle fibers, of course, and metaphase. Okay, what do we? Oh my goodness! Now we've moved to anaphase. Fairly early anaphase because the chromosomes have been pulled apart. The sister chromatids have been pulled apart. They're barely separated. But are these sister chromatids still called sister chromatids? No. Once they get pulled apart, they get promoted to what? They are now full-fledged unduplicated chromosomes. That's what they are, unduplicated chromosomes. So we got a bunch of unduplicated chromosomes going this way and a bunch of unduplicated chromosomes going that way. Okay, we're a little farther along now in anaphase. And so anaphase, we've got these unduplicated chromosomes being pulled this way, these others being pulled the opposite direction. Anaphase. And a little farther along in anaphase here, again, the unduplicated chromosomes being pulled to opposite ends of the parent cell. And this would be probably very late anaphase. And this is probably about as far as they're going to get pulled in the process of this cell dividing. And so, very late anaphase, a whole bunch of unduplicated chromosomes up at this end of the parent cell, and the exact same number of unduplicated chromosomes at the other end of the parent cell. Now, where are we? Well, you see the cell starting to divide. Is that pinching in up there? Pinch, pinch, pinch. Yeah. And so we are in very early telophase. You can almost say we're in extremely late anaphase or very early telophase. And so here's the cell dividing again. Here's the unduplicated chromosomes at each end of the parent cell. And we are definitely now in telophase. We can see the parent. You can see it pinching in over here. We can see it pinching in over here. See, it's pinched in. Here's where the division is taking place. Right here, it's being pinched in. And so we have a parent cell. The chromosomes are coiled up and kind of a wad in the at this end. The new daughter cell. Same thing down here. Chromosomes kind of wadded up in a tight cluster, in the other new daughter cell. And now the cells have just about divided. This would be very late telophase. We'd have a new nuclear envelope forming in each cell, the chromosomes starting to uncoil to where you won't be able to see them. So very, very late telophase.